Hello everyone, this is Ethan from the Huan lab at the University of British Columbia, here to introduce AVIR, software developed by Zixuan Zhang. Often when processing metabolic features, computational problems with integration of chromatographic peaks, as well as misaligned features can occur, reducing precision in quantitative analysis of untargeted metabolomics. AVIR aims to detect these peak integration and alignment issues, automatically recognizing features with problems caused by metabolomic data processing with high accuracy. Using peak intensity in the form of peak area and peak height, AVIR uses a support vector machine model to predict if computational variation exists in features. In this video, we will cover how to download and use AVIR to detect computational variation among features present in multiple samples. AVIR is available for download on our GitHub page. You can check the link in the description below. Additionally, you will also need R and RStudio, or any other program that can be used to run R scripts, such as VS Code. Once you have downloaded and unzipped AVIR, open the R script contained within it in your preferred platform of choice. Here, we will use RStudio. Looking through the script, we can see that AVIR first requires a couple packages to be installed. If you don't have them installed already, open up a fresh R script and type in the following: install packages E1071 and install packages caret. Click source, and once the packages have been installed, they will appear in the bottom right. Next, we will set the working directory. This is where AVIR will find your data and then output the predictions for the alignment of features. Set it to wherever you want the data to be output. Let's take a quick read through of the code to understand what AVIR does. First, AVIR uses the training data file included with the script to test the model to ensure that it is functional. The model is then stored in R. Next, we will demonstrate AVIR's use through a demo dataset included in the repository. Here, we can see we input the alignment table of metabolic features with peak intensity in peak area and peak height, peak area demo, and peak height demo. The format for the tables is included in the README file in the repository. Once these features are loaded, we can see that AVIR first filters the features based on reproducibility in the samples. This should be customized based on your own spectrometer. In the Huan lab, we use a Brucker Impact 2 QTOF, where 1,000 counts are determined as the intensity threshold for metabolic features. The signal of features that do not meet this threshold are discarded as noise. Then, based on the percentage of samples within a feature above the threshold, AVIR will classify it as either a high-quality metabolic feature or low-quality metabolic feature. You can customize not just the intensity thresholds, but also the reproducibility thresholds to suit your metabolomics process. The default settings are as follows. If over 20% of the samples have the feature with 1,000 intensity, the correlation of peak area and peak height will be calculated to generate a prediction from AVIR. For features that do not meet this criterion, they are filtered out and classified as low-quality features, where we assign extreme values of -1 for Pearson and Spearman coefficients as to easily identify these features. Then, to analyze the demo data using AVIR, simply source the script at the top right. It should run quickly and output a file named avirpredictionresult.csv. The contents of this file should show the aligned features, as well as the average RT and MZ, along with the Pearson, Spearman, and other statistical values utilized by AVIR, but more importantly, whether AVIR predicts the feature to have problems from computational variation due to peak integration or misalignment. Using these coefficients, the model predicts the integration quality, which is used to recognize peak integration or misalignment-related problems and then assign a Boolean value, indicating true, referring to no or low computational variation, and thus good integration and alignment, or false, which suggests high variation, and therefore poor integration or misalignment. All low-quality features will be assigned extreme value coefficients and are thus predicted to be false. Through this, you can better understand if your dataset has issues caused by computational variation in your metabolomics processing. To run another dataset, simply remove the prediction output file, add your own peak area and peak height files, and rename the input for AVIR to them. Then click source to analyze the data. This concludes our video on how to use AVIR. We hope that it helped you with your metabolomics workflow.